us on this informational session on the BluVite commercial trials in one of our Lodi AVA locations, vineyard locations. My name is JT Jaeger, and I am the Sustainable Viticulture Specialist with Enartis USA, based out of Windsor, California. Just some housekeeping stuff before we jump in. Generally, the format of our BluVite webinar series starts with a product introduction, followed by a deeper dive into the trial data from 2020 in this instance, although we're presenting on it in 2021. I'll hold off on answering questions during the 30-minute presentation, but we will have a designated Q&A portion at the very end where I'll address your questions. So as questions come up during the presentation, just add them to the Q&A tab, and then I'll make sure to get to them at the very end of the presentation. So this is actually our fourth event this year, covering data that was collected in 2020. A vintage defined by heat waves was done in partnership with the Vintage Report and Fruition Sciences Thibaut Scolache. The Petal & Gap Wine Growers Alliance presentation was in collaboration with Scott Welsh of Jackson Family Wines, and it covers a Pinot Noir trial. The Mendocino Wine Growers event looked at a third-year Cabernet Sauvignon trial in Lake County in the Red Hills AVA. I have added the live links where you can access the recordings, but also you can get access to the slide decks for each one of these events as well. So just very quickly here, I want to talk about the companies that collaborate together. I say cooperative, but really these three companies are essential to make the BluVite technology work in North America and abroad. BluAgri at the top there is our Italian sister company, and they actually create the vineyard product line. They've developed the protocols. They've laid a lot of the groundwork in terms of well, really all the work that we're doing in California is based off of the Italian scientists that work for that company. I work for Enartis USA, as I said previously, and we're distributors of the product in North America, and I run all of our vineyard trials in the West Coast of the United States, but again, based out of the North Coast. SOP actually creates the technology that goes inside the BluVite product line, and they've got a lot of other products that are used in different cropping systems. This company's been around for 20 years, and they've got products in staple crops like corn and soy and other perennial crops as well. But really, more importantly, their venture into vineyards. The reason why we're talking today, and the case that I'm going over, is because they began their venture in vineyards about 6 or 7 years ago in Italy. They also have many peer-reviewed publications in livestock production, but also in, as I said, in staple crops as well. They had a recent publication in corn production, and we're also working really closely with them in BluAgri to get peer-reviewed publications in viticulture as well. So from the map here, you can see that BluVite has a sort of presence on 4 different continents, including North America, Europe, South America, and South Africa. Again, I'm based out of California, and I manage our trials in the Central Valley, North and Central Coast AVAs, as well as our trials in Oregon and Washington State. In Europe, where the product has been developed and available the longest, we have a presence in Italy, of course, where, like where I said the product was created, but also France and Germany. And this is really just all the different grapevine varietals that I've had the pleasure of working on. The ones that I've worked with directly are written in bold, but because we have this global presence, we've been able to work on a pretty large range in terms of varietals, even ones that are relatively uncommon in sort of the new world grape growing. The next 2 slides are a general overview of the mechanisms in which the product works on, and it's really focused on the rhizosphere, or the root influence region of the soil. Both the soil chemistry and soil microbiology is heavily influenced by root growth, respiration, nutrient exchange in this region, right in the rhizosphere. I guess the major disadvantage of being a plant is that you can't move, so I'm not sure they see it that way, but because of this, they have to interface with the soil microorganisms to make a living in the surrounding soils. And so plants are really dependent on the growth of these soil microorganisms, both the bacterial and fungal populations. which possess this metabolic machinery to really access soil nutrition, soil nutrients, really, and especially the ones that are usually minimally plant bioavailable to them, in particular nitrogen and phosphorus. In basically the rhizosphere, there's some very complex interactions that take place between the plant and the roots, or I guess the plant roots and the root-associated microorganisms. And the soil microbial loop is really the reinforcing cycle that results in the emission of these secondary roots, or these fine lateral roots, that are responsible really for the majority of nutrient and water uptake. So these secondary roots are, because of this, very important for the longevity of the vine, especially through extreme weather events like we've seen this last year and even prior, like drought or heat waves, for example, which is pretty much what I would say is the defining factor in terms of, at least in the West Coast of the United States and California, of the 2020 vintage. And it's really because the vine can then better utilize its water and nutrient resources and be more resilient to these events. Microorganisms, like I said, are a critical piece of this loop, which take these nutrients that are plant unavailable, and through various mechanisms, whether it's metabolic or natural population turnover or predation by other microorganisms in the soil, or other organisms rather, and making them available to the plants themselves. And it's really this stimulation of the root microbial interaction in the rhizosphere that the BluVite product line is directly acting upon. We have done some pretty extensive nursery studies, grapevine nursery studies in both California and Italy, to look at the effects of, well, really not only the vegetative growth of these vines, but look at the overall rooting biomass. It's much easier to take a plant out of a pot than it is to dig it up in the vineyard, although we've done pretty extensive soil pits to look at rooting and any changes to the soil environment because of adding this product. But really, this data for this particular case study was collected by my colleague Martina Brogio, and she's essentially runs trials globally for BluAgri. And really this side is the side view of the vegetative growth of these vines within this trial. And this is the top view of those same vines. But really, vegetative growth is great to look at, but we want to see the roots, so we take them out of the pots. And this is the end result when we clean the soil off of them and we actually measure the rooting biomass. We see that there's an increase in the overall rooting biomass. Not only that, but there's higher production of these secondary roots, these fine roots. And as I stated in the previous slide, these roots are largely responsible for water and nutrient uptake. And this has huge implications. So this is kind of when we segue into the actual case study itself. And for this case study, I pulled data from one of our Lodi AVA Cabernet Sauvignon trials to present on today. So moving on from that, this slide lists all of the data that was collected as part of this trial so far, and really when it was collected in the individual phenological stages in which it was collected. I can't really go over all this data. I'll focus in on some certain sample points that will help tell a story as to what's happening in our trials, and ultimately is most interesting for our purposes today. But it's important to note that we will actually have wine analysis from this trial, as the collaborators were able to harvest the respective trial areas and vinify the lots separately. So maybe we'll have some updates on that for everyone who's attended here today. Now we're looking at data that was collected in May of 2020. This data was collected during the period of really early shoot development and rapid shoot elongation. And really this is an important time for overall canopy architecture development, and that ultimately results in the programming of future water needs of the grapevines as well. We had to use an adjacent Lodi AVA trial for this first sample point, but all the other trial data is focused on the one trial example that I mentioned from the beginning. Now, briefly on timing of application, we target the application at a time really that is most effective to stimulate microbial root interactions, which is early season. In early season, this is when most of the uptake of soil nutrients and accumulation of these nutrients are very active into the canopies themselves. And so this is why applications are at bud break and at pre-bloom, so March, April for that first application, May, June for that second application. And it's really so we can influence vine development early on for the rest of the season. Now moving on to canopy observations and measurements. I'll show measurements in the next slides. But generally in vineyard systems, variability of shoot growth is common. And so really one way to understand, at least for our trial purposes, one way to understand the variability of growth during the season is to look at internode spacing, or essentially the distance between two nodes on a cane. In our trial example, this is cordon pruned trial in shoot growth. So the distance between nodes, the internode space, is an indicator of the rate of shoot growth. So internode length will actually vary along the shoot corresponding to varying growth rates during the season. So now I've added red lines to this, and actually these red lines are a standard distance, and it gives you an idea of the distance between the internodes of randomly selected shoots within this trial. And you can see that the treated vines appear to have a more homogeneous internode spacing. You start from the bottom and you move upwards, and they actually have a more homogeneous internode spacing relative to the control. And what this implies is that there's a more uniformly active root system in the treated vines in the early part of the growing season in May. And generally in our trials, there is more uniformity of growth of shoots across the vine in the treated section of the vineyard, with less variation between the growth at any given spur position. And this is kind of shown with the internode spacing, more uniformly active roots, more standardized root growth across the vine. But with multiple years of application, we see that this variability of early season shoot growth is less characteristic of the treated vineyard, meaning there's an increase in uniformity of growth. So I told you we would get to the actual data itself. This is the shoot length data. We also have shoot diameter, which I will not show today. But this is shoot length recorded in centimeters. This is all the data. Has not been cleaned. So basically outliers have not been taken out here. And you can actually see you have BluVite red on the left, you have control on the right-hand side. Just from looking at the data, you can see there's an improvement or an increase in the uniformity, the clustering of data in the treated versus the control. And this equates to a 17.3% increase in average shoot length. And it's not because we have higher extent. The longest shoots are not too much longer in the treated, but it's actually there's a significant reduction in the shorter shoots that brings that average up. And this equates to a decrease in shoot length variability. So a standard deviation of 33 centimeters in the control, whereas it's about a 23.1 centimeter standard deviation in the treated. So now looking at the same data in a more interesting and maybe potentially more meaningful way is to assess sort of shoot quality, basically passing through each measurement through a quality parameter, and then discussing each percentage of each one of these measurements. So a shoot grading of 0 is determined as a non-fruiting or unviable shoot position. Then you move up from there in quality up to 4, which is regular growth. What would be considered regular growth at that vineyard block? And so you can see there was a lower number of an overall percentage of these non-sprouted or weak shoot positions in the BluVite red treated when compared to the control. And then actually more importantly, there's a higher percentage of well-developed shoots. So basically anything above a grading of 3 in the treatment compared to the control. And really what this comes down to is this increased root biomass in the secondary root production that we discussed in the previous slides allows for a higher root shoot growth functional equilibrium later in the season. And because of this, we ultimately see improved ability of the vine to utilize this soil nutrient and its water resource. And this actually manifests in a more uniform and well-developed canopy because of a more active root system. Now looking at our August 2020 sample points, post-veraison is the stage. But by post-veraison, all the vegetative growth should have stopped as the vine should be fully transitioned to the reproductive growth. And I've talked about vine balance in previous presentations on BluVite, but because we see this higher functional equilibrium between root growth and shoot growth, which really also includes mechanisms that facilitate the distribution of photosynthetic compounds, which I'll show measurements for in a previous slide or in a subsequent slide. These photosynthetic compounds are distributed to the different sinks, which include the roots, the shoots, and the fruit. And the fruit ultimately is the analyses we'll cover later on. But overall vine balance in the treated vineyard area has implications for the vine's capacity to fix these carbohydrates created during photosynthesis and accumulate nutrients for growth. And these fixed photosynthetic compounds will be critical for the, like I said, the future ripening, which has implications for fruit quality and yields. So leaf area index is just very generally defined as the ratio of the foliage area to the ground area, but is significantly correlated to some of the things I was talking about in terms of radiation interception, photosynthesis, and then carbon balance within that vine. So it can actually be a good indicator for plant growth, and in turn has huge influence on overall fruit quality, which like I said, I'll cover in next couple of slides. We see a shift upwards from the data. We see a shift upwards in overall average canopy size with about a 26% increase in leaf area index in the treated section of the vineyard. And this is really consistent with our early season measurements, indicating that we have a higher functional equilibrium between the canopy and the roots. And this does lead to a more uniform growth, or leads to more uniform growth, but also more efficient vine transpiration and photosynthesis within a vine, and actually even between vines. And this has huge implications for water stress levels later on in the season, but also the thermal regulation of the canopy, which I think is more interesting. So here's our photosynthetic compound data, the correlated to leaf nitrogen levels. So chlorophyll meters, we use a SPAD meter to measure this, but chlorophyll meters are widely used to guide nitrogen management in other cropping systems and really to monitor leaf nitrogen status over the course of the season. There's a few studies out there that have found that SPAD values actually significantly correlate to leaf nitrogen and leaf phosphorus levels in grapevines. So generally we measure these parameters at bloom and veraison, especially considering that nitrogen, approximately 40% of nitrogen use in the grapevine occurs from fruit set to berry softening. So this is actually after berries have softened and veraison has been completed. In a lot of our trials, we also do bloom and veraison petiole analyses, which I think is a little more effective, but this is quicker and real-time data that I can collect. But similar to what we see in the physical canopy measurements, we also see more uniform average leaf chlorophyll content in the BluVite red treated relative to the control. And this can be directly correlated to higher leaf nitrogen content after the stage in which nitrogen use is at its highest, basically fruit set through lag phase. Post-veraison is also a critical irrigation period for the grapevine, which ultimately determines whether the vines are better able to handle the water needs. And in this instance, we use infrared thermography, and this is actually proven to be a reliable indicator for canopy temperatures. And these images here were taken using a FLIR camera, FLIR infrared camera, and this can actually be a good indicator of water usage in a vine, and actually, more importantly, water use efficiency through high heat. So now taking all of the raw data that the FLIR camera produces, and we've actually done some analyses looking at overall average canopy temperatures. And you can see here, treated on the left-hand side, control on the right-hand side, there's actually a 4.47 degree Fahrenheit average decrease in canopy temperatures between the two treatments. And actually, you can see there's a clustering of the measurements in the BluVite red treated, whereas there's a broader spread for the control. So there's an overall downward shift towards lower temperatures, which ultimately brought that average down. Stomatal conductance is related to the opening and closing of the stomata on the leaves of the grapevine, which is directly related to plant transpiration. So in the data on this chart, we see that there's nearly a 4.8% overall average increase in the rates of stomatal conductance in the treated vines relative to the control. And because the BluVite has on average a larger canopy size with higher rates of transpiration, at this critical phase allows for a higher cooling effect in overall canopy temperatures, so lower canopy temperatures. And because of this, ultimately the vines are better able to regulate their temperatures through these high heat wave events. Okay, now we're going to look at certain fruit quality parameters, including berry weights, Brix, and sugar loading, but also looking at some yield data and how that relates to anthocyanin development. Here are all the measurements, I guess all the analyses that were measured over the season for this trial in terms of cluster analyses, the juice analyses for the clusters. I have bolded the actual data points that we'll go over today, but just know that there's an enormous amount of other data that has been collected, including YANs, assimilable nitrogen, berry volume, looking at even broader phenolic fingerprint panel that includes extractable phenolics and extractable tannins, and then your different acids, malic, lactic, TA as well. So there's an enormous amount of data that's collected. Can't go over all of it today, but this gives you an idea of how in-depth these trials can be for our clients. Now looking at two comparative images from the trial, control on the right-hand side, treated on the left-hand side. And you can actually see the differences in terms of average state of the clusters in terms of dehydration. And I'm going to look at a couple data points to validate these images or the story these images are telling. One of those being sugar per berry by volume. And this component is really a function of berry volume and sugar concentration. I showed this data alongside Brix because Brix is only one piece of the puzzle, one part of the picture of what's happening during ripening, because ultimately phloem transport, once that has ended, then any increase in Brix is due to water loss or dehydration. But you don't get an idea of whether phloem transport has ceased until you have sugar per berry by volume, because that gives you an indication of continuation of sugar synthesis. So is it due to water loss or dehydration, or increase in overall sugars within the berries due to synthesis? So berry weights really are the last piece of that puzzle, and these can be monitored to assess the onset of dehydration. And that's really when average berry weights decrease while Brix increases. But sugar per berry data actually further validates this assessment. And so we'll look at all three of these factors. So now looking at the first part, which is berry weights, and you can see the format here. You have control and treated for both the August and September sample points, and all the rest of the data will be displayed like this. And so you use August, September, and then you have control on the left and treated on the right. But basically what you can see here is berry weights significantly decrease from August to September for the control vineyard, but the berry weights are more or less maintained over time in the treated, a very slight decrease. What that means in terms of percent reduction, for the control, it's 20.3%, but for the treated, it's only 3.4%. And ultimately, this results in overall higher berry weight averages prior to harvest. So now looking at Brix, we can see that in both treatments, there is an increase in Brix over time. Similar increases in both the treated and the control of the vineyard. Ultimately, prior to harvest, Brix maxes out at just about 26 Brix, just under that. And in the treated, we're looking at about 25 Brix, just under that. So moving on to the sugar per berry by volume, this is really critical into figuring out what's actually occurring in the two treatments. And as you can see here from the data, there's actually no statistical difference in sugar per berry by volume over time in the control, indicating that increases in Brix that we saw on the previous slide are not due to active sugar loading, but instead to dehydration. More importantly, we see that synthesis of sugar continues fairly rapidly from August to September in the treated just up until harvest. And so what we see here is a significant decrease in berry weights and a significant increase in Brix with a lower sugar per berry by volume in the control. And this really indicates that dehydration was more apparent in the control area of the vineyard. We also see Like I said, higher sugar berry concentration in the treated just prior to harvest, which implies that overall synthesis of sugar was higher in the treatment. There is this long debated concept in the wine industry that higher yields means lower quality, which can be really an oversimplification, I believe. That is more nuanced, and because of that, it is important for us to not only look at berry weights, like we looked at in the previous slide, but to look at this in relation to other grape quality parameters, such as extractable tannins, extractable phenolics, and really extractable anthocyanins, which is what we're going to look at here today. If you have this higher root shoot functional equilibrium, like I've mentioned in the past, in the treated area of the vineyard, then the vines can better sustain higher yields and ripen fruit at similar, if not higher quality. And so this is also true that the vines may also have a better ability to maintain certain quality parameters over time in adverse weather conditions like high heat. And so we measure these quality parameters to support these conclusions. The quality parameter I'm going to focus in on here is extractable anthocyanins. So anthocyanins are very important for the quality of wine grapes and are hugely influenced by late season environmental factors. They're also influenced by viticulture practices, but generally we keep the canopy management the same between the two treatments in our trials. We can see here that in both treatments that extractable anthocyanins actually decrease over time, but concentrations are consistently higher in our treatment in both time points. So really from this data, it does appear that the ripening potential of the BluVite treated area was higher relative to control, with actually nearly a 10% increase in extractable anthocyanins just before harvest. So now just to sum it all up here, this increased root biomass and secondary root production really allows for a higher root shoot growth functional equilibrium like I've been talking about. And we see this again in the root biomass data from our nursery trials and also the overall canopy growth in our vineyard trials. We see an increase in vine transpiration and small conductance, and this may allow for better cooling potential during high heat events, and this is represented by the thermal images, the infrared thermography, and also the stomatal conductance analyses. There's also this potential for the vines to use more water because of higher rates of transpiration, but actually we see a reduction in water usage, or potential for reduction in water usage in our trials. Most of our trials, almost all of our trials actually, the rates of irrigation, the irrigation scheduling is kept exactly the same. So this has implications for reductions in late season vine water stress, which is actually supported by increased root biomass and also the improved use of water of grapevine's existing water resource. We also, again, in the quality analyses, we also see a decrease in late season berry dehydration and actually higher levels of anthocyanins prior to harvest. And this all can be attributed to the developments in the canopy during the season. This now brings